All right. So, delicious Doritos. So we just finished talking about this, yeah? Am I starting too soon? No, okay. So, so there's a few other things I want to talk about briefly. I think this is kind of interesting. How do we classify, what constitutes a species in eukaryotes? Are horses and donkeys related? Are they the same species? Why not? Oh, yeah. So I don't know where the trash can is. So, horses and donkeys, yeah. Miriam. Ah, not quite. The mixture of the two species is the horse and donkey. When they form a mule, yeah? So a mule. So. Yeah, so. So mules, generally, the number of chromosomes in a horse and a donkey are slightly different. I think one of them's got 44 and the other's got 42 or something like that. Yeah. And you better not quote me on those numbers. But because they have slightly different even numbers of chromosomes, when a when a mule is formed, it has an odd number of chromosomes. And when meiosis happens in that donkey's eggs or sperm, and I guess, I don't know, a male horse and a female donkey is a mule, and then a female horse and a male donkey is a, there's another word for it, but I don't remember the word. Yeah. Anyway, so there's an odd number in that horse donkey cross creature, whether it's a mule or the other thing. It's got an odd number of chromosomes, and so when it does meiosis, one of those chromosomes gets lost, and that's part of the mechanism of why it's sterile, yeah? So, so what about bacteria? So are they the same species? We say they're not because they cannot be mated together to reproduce and have a fertile offspring. Yeah? Can a polar bear and grizzly bear mate together and produce fertile offspring? They can. So are they the same species? From the biological standpoint, we would say that they were the same species, yeah? There are other criteria, right? Are those do those species repeatedly reproduce? And that is, are they in reproductive isolation, yeah? If a grizzly bear and a polar bear never are separated by the Brooks Range, then maybe they never encounter each other unless a zookeeper hooks them up, yeah? That's another definition of a species, yeah? But as far as bacteria are concerned, yeah, do either of those definitions fit with bacteria as far as what a species of bacteria is? No. Why not? They reproduce asexually, yeah? If a species reproduces asexually, then how can we ask if they can be mated to make a fertile offspring, yeah? They don't they generally don't mate to reproduce, or they never mate to reproduce. Is that, you guys get that? So, for bacteria, we just say it's a population of cells with a similar set of characteristics, yeah? Good? So, we've already talked about the three-domain system. We talked about the endosymbiotic theory, where mitochondria and chloroplasts formed ancestral eukaryotes. Let's see. Bacteria and archaea are evolutionarily different from each other. Prokaryotes are the most ancient things on Earth. We can see there's fossils from billions of years ago that are structurally very similar to structures we can still find today. There are several kingdoms of eukaryotes that we've already talked about. We know the endosymbiotic theory that describes the origin of eukaryotes. Here's something that I thought might be interesting, that we talked about the endosymbiotic theory. Many protists have what are called secondary and even tertiary endosymbioses, yeah? So, have a eukaryotic protist, unicellular, and it swallows up another eukaryotic protist, and that other eukaryotic protist starts living inside it, and you have a euglena, yeah? A euglena is a little creature whose ancestors, not 2 billion years ago, but maybe 500 million years ago, swallowed up a eukaryote and had that eukaryote take up residence inside it for evolutionary time, yeah? Dinoflagella, kelp, malaria. These are things that have multiple endosymbioses in their history. Isn't that weird? It's interesting, isn't it? Anyway, and if you look at these guys, how many nuclei do they have in their cells? They would have 2 if they have a secondary endosymbiosis. And they're asymmetrical. They're not the same. One is like a big nucleus, and the other is like this little bonus nucleus, yeah? Like a little extra nucleus. Okay. So, what about viruses? How do we classify those? Again, they're not alive. And furthermore, if we look at their DNA sequences, what we find is a lot of times they'll pick up DNA from the species that they infect. And they often bear more DNA sequence similarity to their hosts than they do to each other, yeah? Viruses are not one thing. Viruses, right, life has evolved once on Earth and diverged from there. Viruses have not. Viruses, it is likely, evolved independently many different times. And viral genomes are weird and diverse, and we'll talk about them very shortly. They can be double-stranded DNA like our genomes can, but they could also be single-stranded DNA, right? Viruses can have DNA that's just one strand. They can have RNA genomes. They can have no DNA at all and still be an effective virus. HIV is an RNA virus. Many of you who did presentations about viruses learned that they were RNA viruses. Raise your hand if you found that you were doing a presentation on an RNA virus. 1, 2, 3, 4. Yeah, so 4 of you learned that on your own, right? That's something we never even talked about, and then you had to learn that on your own. So good on you if you found that out. So RNA can be You ever heard of double-stranded RNA? Well, we know that RNA can bind to itself. When we see a transfer RNA, it forms little loops and so on. But here we have RNA positive or negative. What's that about? Any guess? It's not electric charge. What does that mean? Anybody know? Anybody guess? So plus and minus in RNA is effectively thank you. Sense and antisense. Do you know what that means? That means this one is effectively an mRNA, yeah? That codes for what? The viral proteins, yeah? The other one is antisense, the opposite of an mRNA, yeah? We'll talk more about that. Or does anyone think they get it? Yeah, so the sense one is effectively a messenger RNA that's ready to go, ready to be grabbed by a ribosome and turned into viral protein, yeah? Or used to make viral protein. Does that make sense? The antisense is a little different. Antisense is, do you remember the codon chart? No, don't stop recording. Let's pull up a codon chart, shall we? There's the dentist that spread HIV. So images, codon chart. All right, so here is our nice little codon chart, yeah? Imagine I've got a, can you guys see this? Maybe I'll make it a little bigger. Is that big enough? Can you see that? Ugh. Not if I minimize it. There we go. All right, so there it is. So imagine I've got, give me a pen here. Let's take this little sequence. We'll go AUG. What's that mean? That means start, yeah? Then we'll go AAC, and then we'll go CGC, and then we'll go UGA. Got it? So this is start. This is AAC. What's that? AAC, asparagine. And then GCG, alanine. And then UGA is stop. Yeah? Right? So I've got start, asp, ala, stop. Yeah? Does that make sense? Well, A always matches with U. U always matches with A. G always matches with C. A with U, A, U, G, G, C, G, A, C, U. Yeah? So, what do I have here? This is a messenger RNA. In other words, the plus strand. What's this one? This is the complement of the plus strand, yeah? And so that's the minus strand. Right? What does this code for? Well, effectively, it's gobbledygook. What do we got? UAC, which is tyrosine. We don't have a stop codon. No stop codon anymore. We don't know that we're supposed to start. Or no start codon anymore. AAC, what's that? Asparagine. GCG, what's that? GCG. Alanine again, so that one's the same, strange. And then ACU, what's that? ACU 309. Right? And yet, right, this is a bunch of amino acids that have nothing to do with what we're trying to make, right? So if this is gobbledygook, then what? Well, this doesn't make any sense on its own, but it can be used to make this, yeah? If this was my genome, then all I would need to do is transcribe it effectively to make a messenger RNA out of it, and then I'm ready to go. Does that make sense? You guys get that? So this doesn't code for anything itself, but it does code for the genome of the virus, or the right? It is the complement of the proteins of the virus. Yeah. Okay. So that's what's meant by negative strand. Where was I? All right. Antisense. You know what I mean? Antisense. Get it? Okay. So moving on. What else do we have to say? A bacterial species. What is it? D. A person with similar characteristics. That is correct. Good. All right.
So a lot of the slides that you went through aren't on this. Are you going to do another version of it on?
Oh, I should post another version if that's not okay. Sorry about that. Okay. Say classification versus identification. What's the difference here? What did you do in lab? Which did we do? We did identification, yeah? Right? And once we identify it, then we can look up its evolutionary history and find out how to kill it, yeah? Does that make sense? So identification is going after an unknown, yeah? Is that important? Sure, right? Again, the patient doesn't care about the history of the organism. The patient just wants the organism to die. Yeah? If it's an infection, we just want to kill it, yeah? Right? Does classification matter? Sure. The more we know about the evolutionary history of things, the better we're able to kill things when we find them, yeah? Right? So how are they different in their mechanisms? Well, we can do, when we do identification, we can end up making something like a dichotomous key that is really good for identification, but not necessarily for phylogeny, yeah? So, is it gram-positive or gram-negative? Well, if it's gram-negative, does it ferment glucose? If it does and it only forms acid but not gas, well, maybe we've got Plesiomonas shigalloides, yeah? And if it doesn't, well, if it forms gas along with acid and it's motile at 37 degrees Celsius, well, maybe we've got Aeromonas, yeah? Guys understand? You understand what's meant by dichotomous? What does di mean here? Two, right? A dichotomy is yes or no, Coke or Pepsi, et cetera, et cetera. Does that make sense? So a dichotomous key is a bunch of yes or no questions. And effectively, each of those tubes was, or usually is, a dichotomy, yeah? There are those like the TSI that gave you multiple questions, yeah? But most of the time it's dichotomous. Positive for urease or negative for urease. Positive for citrate or negative for citrate. Positive for indole or negative for indole. Got it? Okay. So, Yeah, there's another one. This is something called an enterotube that's used to test. You can see effectively this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 little differential auger plates, yeah? Only they're not plates, they're in this little tube, but there's agar in there and there's media inside it. And if we take some bacteria and put them on the end of this metal rod, we slide the rod through the tube and the bacteria inoculate. As the rod is slid through, the bacteria are inoculating each of these chambers one by one by one, and then we can see how they grow and what they do, yeah? Looks like this is urease positive, citrate positive, lysine negative, ornithine negative, etc., right? And then we can add it all up and we can say, oh, we must have Proteus mirabilis, yeah? Okay. We can also use serology. This is where we do what's called slide agglutination, where we treat a bacterium with antibodies to see if the antibodies bind. Antibodies, as we'll find out later today, are super specific, yeah? And so we can actually use antibodies found in serum effectively in the blood plasma of hosts that have been infected with this bacterium. And these antibodies are so precise and specific that they will tell us one strain of bacteria from another strain. Antibodies are more precise than species. They can go down below the species level to the serotype level. So when you see a serotype is a subtype of a species where some serotypes are super pathogenic and others are less so. Okay. We can do identification with DNA analysis. We can look at GC content. This is kind of strange. We won't be talking about that too much. And then we can do DNA fingerprinting that we talked about before. Could we use this to identify bacteria? Sure. Purify out their DNA and do restriction digests on it. We should get the same thing as if we did restriction digests on people and their DNA to identify DNA at a crime scene. Yeah. This is another technique called nucleic acid hybridization, and this is a technique that is already in use, and you would benefit from understanding. Yeah. So, organism A versus organism B. How closely related are they? Well, one way to figure out how closely related is by heating up their DNA, mixing it, and seeing how well it sticks. You understand? So, here are 3 hypothetical outcomes. Outcome A, B, and C. Which outcome suggests that organisms A and B are the most closely related? A, right? The one on the left shows the DNA sequences are completely identical. Because they're completely identical, all the bases match up and the 2 strands of DNA stick together. Does that make sense? Well, what if the species are unrelated? If the species are unrelated, then the DNA doesn't stick together at all. And if it sticks together a little bit, well, they're closely related, but they're not the same. All right, so how do we use this? Well, we can use this to do something called fluorescent in situ hybridization. Fish, yeah? Hybridization, you understand that? Hybridization is this, getting the 2 strands of DNA from 2 different things to stick together. Yeah? So, what if we took organism B's DNA and painted it green? Yeah? Well, if we did, what would this look like? It would look green. Do you understand? Shall I say that again? If I take organism B's DNA and spray paint it green, and then I take organism A's DNA and mix it together, yeah, what does this look like? Is this green or not? Yes, it's green. Is this one green or not? This one won't be green. You understand? So, how do we identify Staph aureus? Well, you take a sample from somebody. Somebody's got an infection. There's pus in there. It looks nasty. You wonder if they've got MRSA or some kind of Staph aureus, right? Do they have Staph aureus in their infection, or is their infection something else? Well, one thing you could do is take a swab from that wound and put it onto an MSA plate and see if it grows, yeah? See if it's positive for mannitol, and if it's positive for mannitol, the plate looks green, or it goes from, it goes from pink to yellow, and you can tell that that bacterium is eating mannitol. Does that make sense? And Staph aureus eats mannitol, and if it doesn't grow at all, it's not Staph aureus because it's too salty for them, yeah? Guys with me? All right. So, how long does that take? A while, yeah? It takes a day, yeah? And maybe the patient's got a really nasty infection, right? So maybe you want to find out sooner than that, yeah? So what else can you do? Well, you can do this, fluorescence in situ hybridization. What do? Take the patient's wound sample, instead of smearing it onto an agar plate, smear it onto a glass slide for the microscope, yeah? Heat fix it. Fix it onto the slide, yeah? Then get some DNA from the fridge that is Staph aureus DNA that somebody took out of the Staph aureus, fluorescently painted red, and then put it in the fridge. You understand? Right? So, somebody in a lab in Cleveland or wherever gets a bunch of Staph aureus, they purify out the DNA, they tag the DNA red. Yeah? They put it in a bottle and sell it to the hospital. And the hospital gets it, puts it in the fridge, and now we've got this patient that we're worried has a staph infection. Yeah? Get the bottle out of the fridge. Put a little of this spray-painted red DNA onto the slide that we just fixed with the patient's sample. Guys got that? Guys with me? Yeah. Thank you. So, now what do? We try to wash it off. Yeah? If we have Staph aureus, will it wash off? No. The A's will bind to the T's, and the C's will bind to the G's, and it'll stick like glue. Yeah? And if it ain't the same species, then what will happen? Those fluorescent pieces will just wash right off because they won't stick. Does that make sense? All right. And what do we do next? We put it under the microscope. And if we see red, what do we know? There had to be Staph aureus in there, because if it wasn't Staph aureus, it wouldn't have stuck. Does that make sense? You guys got that? All right. Powerful? Powerful and rapid, yeah? We can know this in an hour instead of 24 hours, yeah?
So, the heat fixing with the heat component,
would you need to heat it? What breaks the hydrogen bonds between the Staph DNA? Yeah, heat breaks hydrogen bonds. So the heat breaks the hydrogen bonds, and so the DNA separates, and some of the fluorescent DNA can tag in there, just like in PCR. You would need to heat. You would need to add some heat. Yeah. You would probably this fluorescent DNA would probably be in a liquid solution. You'd put that on the slide, and the liquid solution would be heated up to nearly boiling. And as it got heated up, the hydrogen bonds in the patient's DNA or the patient's infections DNA, rather, and that DNA would separate just like it does in PCR, and then as it cooled, some of the fluorescent DNA would stick in its place, yeah? Does that make sense? Excellent, excellent, excellent. All right. So, gene sequencing. We've talked about that, right? We can look at DNA samples and identify bacteria, yeah? We talked about how knowing the human genome would be useful for patients, yeah? Would knowing the DNA samples from bacteria? If we can do DNA sequencing in a few hours, why not just DNA sequence every sample we get, yeah? Right? Right? If we did that, we would not just know that we had Staph aureus, we'd know that we had MRSA. We'd know we'd have which particular kind of MRSA, yeah? Does that make sense? With DNA sequencing, we know as precisely as science knows what species of what bacteria we have where we've got it. Does that make sense? You guys understand that? So DNA sequencing is certainly the future of diagnosis for many diseases, is already the present for things like flu, yeah? If somebody hawks a loogie into a cup and they take it down to a lab at MGH, they can take that and put it into a DNA analyzing machine for flu and tell you whether you've got H1N1 or H5N1 or bird flu or swine flu or whatever the heck flu, or whether you have no flu at all. Yeah? By looking at the DNA of whatever's in your sputum, they can find flu viruses if you've got them, and they can find cold viruses if you have those instead. Does that make sense? And they can do that now. Yeah? Those machines exist now. The machine that does DNA sequencing for bacteria exists now. I guess the next step is streamlining that such that we can get the DNA sequence for every bacterium that might be in somebody's surgical wound site or catheter infection or whatever infection we're curious about. Does that make sense? Right? All right. So that's chapter 10, yeah? I want to talk a little bit about chapter 13 if I can, viruses, and then we'll do 16 and 17 after lunch, unless you guys want to take lunch early. Lunch early, or let's just do this? Okay, so I'm going to try to bang out really quickly the thing that I think is essential I'm going to try to complete discussing chapter 13 very quickly, yeah? Okay, so, and I think this thing is done.